Okay, so I want to tell you guys a couple of things before we get started, and then we'll do urinary system today. And that is, you're going to have the opportunity for 5 points bonus on your second lowest test, because if I put it on your lowest one, it might get dropped, right? Okay. Even if you don't need the bonus carry, do it anyway. Because it's just a survey, and basically over the last year we have been doing a success and retention initiative with human anatomy to try to figure out how our student population can be more successful in anatomy. Anatomy nationwide has a 50% drop, withdraw, and fail rate. Ours is way better. We're like 32%, but we want it to go up to go down below 25. We want to get like about 70-75% of our students passing the first time. And so we're working on it. And so last year, this time, I gave this survey and assessed my students stuff like, I'm not gathering personal information about you, like I'm not going to ask you to put in your name, but demographic information, like if you're a first-generation college student or if English is your second language and it's not the primary language spoken in your home, that matters. Our student population matters. How much you work, right? That kind of stuff. Also assessing what kind of barriers people encounter to success in anatomy. And some of them can't be helped, right? There's too damn much information. It's not like I don't know that, and it's not like we can do a damn thing about it. What do you think I can do about the amount of information in anatomy? Literally buckets. That's what I can do about it, right? I can just make sure that you've got all the resources you need to do well. So asking you about your barriers to success, and what came out of that is some stuff that we thought would help. Last time we did it, you guys told us that it's really, really difficult for you when lecture and lab are not on the same page all semester long. So we gave that information back to the lecture instructors and said, students say that as much as you possibly can, lecture and lab need to be together, right? And so we're going to see if that wiggled anything. I got even better at putting them together after that. And then also some of you guys said, well, my work schedule, I can't do it unless there's like a Saturday open lab and an early morning one or a late night one, so we put those in for you. So I'm going to give you guys that opportunity to do that. And if you will do it, it will help us to continue to improve the experience for students. Because although I realize this is difficult, I hope you also realize that there are people who are putting a lot of work into making it possible for you to be successful. So we do want you to. It's not like everybody's going to be successful because a lot of people don't have the time that's required to get through this. But anyway, that is one opportunity. And the next one I'm not sure about yet, but I'll tell you about it just so that you can clear your schedule if it's important to you. The next one will be a week from Friday. We are hiring 1 to 2 new faculty members from the biology department. And because we are a teaching institution, the last step of hiring is always to take the top candidates and have them do a teaching demonstration before students. And then we ask you what you think, which one you would want to take classes from. And I'm hoping that we will have one faculty member for A&P that we're trying to hire. I'm trying to make it so that they're going to do a urinary system lecture so that not only will we benefit from you giving feedback, but you'll benefit from the review, right? And there'll be one who's interviewing for freshman biology, zoology, botany kind of position, like that one. So that'll be next Friday, I think, and I don't know for sure, but I'm telling you so that if you need the points, you can clear your schedule. It will be on the hour from 9 o'clock to 4 o'clock, okay? You don't need to go 9 o'clock to 4 o'clock. I would love it if you could possibly go to 2 so that you would have a comparative, right? You would say this one more than that one, right? Does that make sense? And then we take your information, and as we're figuring out who to offer it to, the student evaluations come into play. Go ahead.
Did you say on Redwood campus?
It's going to be on Redwood campus. I don't know any more details yet. Okay. I think the A&P ones might be in the afternoon, and the freshman biology ones might be in the morning. But I'll tell you when I find out, and I'll post it on Canvas. And I will also post that assessment, that retention survey on Canvas, probably post it today. So you can go ahead and get it out of the way if you want. It'll only take, I think it average 4.5 minutes or something.
How many points
right?
are you offering for this?
5 points for the success and retention survey. I don't know about the other one yet because I need to figure out what's happening, right? Go ahead.
Can we do both and get points?
Yeah. Mm-hmm.
Oh.
Yeah. Yeah. You can do both. So I think it'll probably be, if you do both, about 8 points total, which will be pretty good, right? pretty good for you. So you make your own choices. Do not complain if you got shit to do on Friday. I can't change people flying in from other states for your schedule, okay? Just clear your stuff, that's all I'm saying, okay? All right, so now we're doing urinary system. With urinary system, okay, so it seems like it's boring initially, but it's really not. It's actually pretty damn fascinating. So what you have known since you were a little kid is, of course, the urinary system produces urine, right, which is waste products, but that is not all it does at all. Because everything that ends up in your urine came from your blood, right? So if you, for instance, decide that I have too much calcium in my bloodstream, the urinary system probably can take care of that, right? If I decide that I had not enough glucose, then the urinary system can take care of holding on to it and make sure you don't get rid of excess. In addition, since everything that goes into your urine came from your bloodstream, then can you see how the urinary system is intimately related to blood volume and therefore blood pressure? Right? If you pee it out, then what happens to, if you pee out more water, yes, what happens to your blood volume? It drops, right? Because it came out of your blood. And what happens to your blood pressure? It drops, right? So although you may not realize it, the urinary system is one of your major ways of controlling your blood pressure. Yes? And so you can't talk about urinary system, can't talk about cardiovascular physiology without talking about urinary physiology. So it's really intimately related, and it's super important. We'll kind of get that sorted today. In addition, the kidney also releases some hormones. One of them, your kidneys are the one that gets to decide when your bone marrow makes red blood cells. So your kidneys actually release a hormone called erythropoietin, or EPO, that causes erythropoiesis. It causes you to make red blood cells. So your kidneys are regulating both what's in your blood and also how much of it you have. So there's like an intimate relationship between your cardiovascular system and your urinary system. Okay, so we're going to get into a little bit of the fizz today. Not hardcore fizz, just a little bit of the, this is why you give a crap fizz. Go ahead.
What was the hormone called one more time?
Erythropoietin, EPO. Okay, so we'll get into the basic anatomy. Actually, I want to do something different first. I'm going to get into the physiological basis, then we'll do the basic anatomy. Okay, so I'm going to start with this basic understanding. This does not represent the anatomy. This represents sort of the physiological flow. So basically what's going to happen at the kidneys is ultimately we are going to get to the point where I have microscopic blood tubes, capillaries, yes, microscopic blood tubes, right next to microscopic pee tubes. And whenever you have microscopic sets of tubes next to one another, there's just kind of like the placenta idea, right? You can have exchange between them. You've got a whole bunch of surface area, you have a whole bunch of opportunities for exchange. If you're really porous, it even helps more. Does that make sense? Okay, so what I'm going to get to, and we'll get to this point at the kidney, is ultimately the blood coming into the kidney came from what artery? Duh. Renal artery, right? Okay, came from the renal artery, and ultimately the blood that's leaving the kidney is going to go into what vein? Renal vein. You with me so far? Does it make conceptual sense to you that if the kidneys are doing what I said they're doing, the blood in the renal artery will be different than the blood in the renal vein? Yes? Different in volume, different in composition, different in waste products, yeah? It's going to be different, yes? Okay, so this is of course circulating, but everything that came out of here is going to end up in here, right? This is not circulating. Urine does not circulate back into your body. It starts at a blind end in the kidney and it ends up in the toilet. Yes? Okay. Everything that ends up in the toilet, you consider that it was excreted. Yes? Okay. But there is a 3-step process to make urine and cleanse your blood. Okay? I want to introduce you to these terms because renal anatomy is really fundamental for understanding renal physiology. These two knuckleheads right here took phys before anatomy and made their lives difficult. But if you take anatomy first, then you're going to know all this terminology before you start trying to understand renal physiology. I mean, they did well, so they're not knuckleheads. But it's not the easiest way to do things. Okay, so here's the metaphor I want to use. Okay, your blood, let's say you got super dirty blood before it goes through your kidneys. So I want you to use the metaphor of cleansing your blood like cleaning a hoarder's house, not whorehouse. hoarders. I mean, that could be going on in there because we don't even know, right? Okay, so here's what I'm gonna do. I'm gonna do a 3-step process to clean this house. The first step is really going to be, there's so much crap in here, I'm not even gonna look at it. I'm just gonna pull everything that will fit through the door out onto the lawn, right? Okay, this first step is not selective except by the size of the holes, and it's pressure driven. Do you guys remember what transport mechanism is pressure driven? What did you say? Not diffusion. I'll give you 3 choices. Diffusion, osmosis, 4 choices. Active transport, filtration. Filtration is pressure driven. Okay, so, ready? So you guys tell me, if it's pressure driven, it's going to go from high pressure to low pressure. Which of these fluids is higher pressure? Yeah, right? So filtration is going to go this direction, yes? What are you going to filter out? Anything that will fit through the door, yes? Which means that I am going to dump out onto the front lawn, not the furniture, that's whole cells, right? But cat boxes with cat turds in them, 747 cans of corn, grandma's china, box of money, all fit out the front door. I'm not selective, right, except by size. Why the hell did I do it? Well, I can't even start until I've got like crap out of this house, right? So I'm like putting it all out on the front lawn. Does that make sense? So this is filtration. So what do you filter out at your kidneys? Well, you're not selective. You filter out water, about 47 gallons a day, which means, you're gonna have to reabsorb a hell of a lot of it. You filter out ions because they're small, right? Yeah, and water soluble. You also filter out nitrogenous waste products like urea and uric acid and ammonia. Does that sound familiar? Nitrogenous waste products. That's how you get rid of most of them in urine, okay? But you also filter out good stuff like grandma's china, like glucose, amino acids. Those are money, right? So, we dumped all of this out onto the lawn. Obviously, we're not finished with the house, right? Because what we are going to do next is we're going to say, okay, we got it all out on the lawn, and now we need to select what I'm going to pull back in. And by select, I mean that you are going to use specific carriers, specific channels that are activated to allow transport of things. So this was not selective. The next step is going to pull stuff back in, and it is selective. Does that make sense? This step is going to be called reabsorption. Which direction is it going to move stuff? Yeah. Okay. What is it going to move? Well, obviously you don't pee 47 gallons a day, right? So obviously you're going to move a hell of a lot of water back in. Does that make sense? Okay. How about waste products? Do you think that I generally bring the cat turds back into the house? Just leave them out there, right? Okay. Leave the damn things out there. How about grandma's china? Bring it back in. How about the box of money? Bring it back in. How about the corn? Well, I don't need 747, but I might need 10, right? So here's what you do. You bring back in water. You bring all the glucose if you can, and amino acids, ions, as needed. So if, for instance, I ate a 1-pound bag of Doritos yesterday, I got a boatload of sodium in my system, right? I push it out, right? But I've still got plenty of sodium in my system. So can I leave it out there, right? So I don't need to reabsorb sodium. But what if I hadn't eaten for a little while and I was calcium deficient? Yeah? Then I push it out because it's small enough to fit through the holes, but I bring it back in because I need it. Does that make sense? Okay? So, and the question that may come up is, why the hell do you push it out? Well, because you can't control pushing it out. It just goes if it's water soluble and fits through the holes, right? Okay, but then the last step is critical evaluation of the house, opening all the cabinets, looking under all the beds, finding that basement in the bathroom that has 800 rolls of toilet paper stored in the shower, right? Okay, finding all of it, and then you do selective secretion. In which direction do you think secretion is going to go? That direction, yes? Okay, what are you going to secrete? Primarily, any turd box that you didn't find before, so any waste product that didn't go out, and they don't go all out, right? Because it's just whatever fit through the holes that was right there, right? You don't get rid of all of it just by filtration. You have to sometimes select it and move it. So this is selective secretion, waste products, and then anything that it had excesses of, right? Does that make sense? So wastes and excesses. So in this situation, ate a 1 pound bag of Doritos by myself, right? I was depressed, don't judge me. Filtered out a boatload of sodium, reabsorbed 0 amount of sodium, because I've still got a bunch in my bloodstream. And then I get to this step. Do you think I'm going to try to secrete some more sodium if I can? Yeah, right? Try to secrete some more sodium. Does that make sense? Go ahead, Carrie.
Is that active transport?
If necessary. You will do active transport if the concentration gradient doesn't favor it. But the body always does things the cheapest, fastest way it possibly can. So if you can do something passive, you will. But generally speaking, if I already had a bunch in the urine, I would have to pump sodium into there. Does that make sense? Yeah? Just because of concentration gradients. Okay? So what happens here is that by the time I get finished, I hope that I have reduced my level of waste in the bloodstream. I hope that I have homeostatically regulated my level of ions in the bloodstream. Yes? And I hope that I have saved all of the nutrients that I possibly could. Yes? Okay? And I always lose water in the process. Always. Right? Because the only way to get rid of water-soluble stuff is to lose some water to get rid of it. Yeah? So I always lose water in the process. So, of course, this is definitely going to be lower volume, but it's also going to be a different composition. Good? Yes? And, of course, this is going to be a different composition depending on what you ate. Breakdown products, all that kind of stuff, whatever you had excesses of. Good? But it's generally going to be relatively high in waste products like urea and uric acid and ammonia. That's how you get rid of them. Good? So the equation, it's not really like a hard equation. It's just excretion, yes, equals, see if this makes sense to you, what you filter out minus what you reabsorb, right, plus what you secrete. Does that make sense? That's what you end up excreting. Good? So that's the phys basis. So today what we're going to do is we're going to get all of the gross anatomy until we get to this microscopic anatomy. You can't do this until you get to a capillary, right? So we got to get all the gross anatomy until we actually get to the capillaries, and then we'll talk about this. In phys, you learn way more details, right? But you spend almost all your time right there. You don't do much of the gross anatomy. You spend almost all your time talking about how to control filtration, reabsorption, secretion. Good? Okay, so let's do the easy part, which is the gross anatomy. Gross anatomy of the urinary system is just freaking easy as pie, if you ask me. These are kidneys. These are ureters. Yes? That's the urinary bladder, and that's the urethra. Most common gross anatomy mistake is to mix up the term ureters and urethra, but think about it. One of them sounds wrong if you say it plural. Yeah? Because you ain't got 2 of those, right? Urethras sounds dumb because you do not have 2 urethras, right? So you got 2 ureters, 1 for each kidney, right? Urinary bladder, urethra. Good? Okay, so the kidneys, as far as the location, the gross anatomy, where they sit, they're in the lumbar region, right? But one of them's slightly lower than the other one. The right one's slightly lower. Does anybody know why? There's a big old chunky. Yeah, the liver is a hog. The liver is way bigger than you expected it to be, right? Okay? It's just way the hell bigger. So the liver is on the right side in the vast majority of us. Although, did you read that article this week about the woman who lived to 100 and had all situs inversus?
Crazy.
Okay? Yeah, there's one of those at the body donor program too, in which they have all the organs flipped. So in most of us, the liver's on the right side, so that pushes the right kidney down a little bit, right? That ends up manifesting in everything being pushed down. If you were a dude, you probably haven't noticed. Well, maybe you have. I don't know what the hell dudes notice. But it usually means that your right testicle is slightly lower than your left testicle, which is important because you don't want the clap going, right? Okay. If, however, you have situs inversus, a lot of times the first way that men are diagnosed with situs inversus is when they're actually doing a physical exam. And the doctor's like, hey, I think your heart might be on the other side. And you're like, did you not pass med school? That ain't my heart. No. But because if your left testicle hangs lower, it usually means that you got some flip going on. So most men, the right testicle hangs lower. So they're not actually together. In women, you would just have to find out, I don't know, anecdotally, but that woman, they didn't find out until she donated her body. She was a cadaver donor for anatomy, and that's how we found the other one too. It's interesting when they have situs inversus, the one that's at the body donor program, they found it because, of course, the med school does first anatomy, right? And you don't know what's inside the body before you open it up. It's like a freaking Cracker Jack box. It's so damn fun. So I don't even know what I'm going to see. It's fantastic. So they opened her up, and the med school students are like, I thought that, and then they immediately take it away from them because they didn't want for you to be a doctor. Dr. Jasmine, who from the beginning of her career has the impression that the heart's on the right side, is going to be Dr. Confused Jasmine. So they just took it away from her immediately, right? And every single time she has to flip everything. So anyway, let's talk about the kidneys as far as their position in the body. All right, so this picture is a little hard to understand from there, so I want to go ahead and tell you that you're probably going to have to look at it in your textbook just because it's a little hard to see what you're looking at. But I want to talk about how the kidneys stay back here. So when they're in the lumbar region, right back here, do you remember retroperitoneal? Do you remember that concept? No? From the beginning of the semester, they're behind the parietal peritoneum. So instead of the like the organs in the ventral body cavity, most of them are wrapped with their own layer of visceral peritoneum and then hooked to the wall by a mesentery, and then the wall is wrapped with parietal peritoneum. So the kidneys are retroperitoneal, so they're behind the parietal peritoneum. They don't have a wrapping of visceral. Does that make sense? Okay. So they're smashed back there. That means that you're going to have to sort of wedge them back there because they're not hung to the body wall by a mesentery. Okay. And so they're smashed back there, and primarily what you've got is packing material around them, fat, right? And then a couple of different layers of packing tape holding them onto the back, right? So we're going to look at that relationship right now, how they're actually back there. It's not really that bad. It's just a little hard to see from this far away. Okay, so it doesn't matter which kidney you start with, but this one's labeled, so we'll do this one. The outer boundary of the kidney is called the fibrous capsule or the renal capsule, and it's dense, irregular connective tissue. It's just like the boundary of the kidney. But then superficial to that, you're going to have your first layer of packing peanuts, which is fat, right? Ready? So this first layer of packing peanuts around it, the fat, is called perinephric fat. Yeah? Literally the fat around the kidney. Good? Okay. Then I'm going to have a layer of packing tape, right? You with me? So this layer of packing tape is this layer that you're seeing right here. Do you see it? That is called the renal fascia. So imagine that you've actually got some connective tissue that's just holding that fat against the body wall and the kidneys. Okay? Then you're going to have another layer of packing peanuts. Do you see this? Yeah? That's called the perinephric fat. It's a little further away from the kidney, right? And only superficial to that will you have your last layer of packing tape, which is the parietal peritoneum. Yeah? Okay, so remember back at the beginning of the semester we said that the fat around the kidneys was actually wedging them back there. Do you remember us talking about anorexia and losing weight? Okay, so one of the things that can happen if you lose weight super quickly, like you have some really terribly aggressive disease, like a terribly aggressive cancer or whatever, it makes you lose weight just really quickly. Or if you have anorexia and you like get super duper thin, what will happen is you'll lose all the packing material, right, around your kidneys. And your kidneys can actually slide down between the body wall and the renal fascia, and they'll just slide down. When they slide down, because of this anatomy, you can kink your ureter. Okay? And here's the thing that you need to know about the kidneys. They never stop making urine. If you can't get it out, it's just going to back up into the kidney, and they're going to be basically bathed with waste products and improper pH, for instance, and it will cause kidney failure, or can cause kidney failure. So I don't know if you're aware of this, but one of the leading causes of death in people with anorexia is kidney failure. As you lose too much fat, and they just slide right back down there, and then one of them dies or the other one dies. Does that make sense? Okay, so that's kind of how they're packed in there. So now let's look at a coronal section of the kidney and start learning the basic anatomy. So one of the good things about the kidney is that the picture looks kind of similar to actually what the kidney looks like if you look at a coronal section of the kidney when you're in lab. So for instance, it is lighter on the outside. It does have these darker triangles that you can see going through it. You can see the ureter attaching to it. Unlike the brain, in which you're like, sure, whatever. That region, sure, I'll call it that. I don't know what the hell you're talking about. But the kidney does look kind of like this. So basically when you're looking at the kidney, and this is a coronal section of the kidney, it has just below the fibrous capsule around the outside, you're going to run into the cortex and then the medulla. Every single time you have a cortex and a medulla, which one's superficial? Cortex, right? And then the medulla is deep. And you're going to run that into that in the kidney, and you're going to run into it in lymphatic vessels. You're going to run into it in the ovary, in the brain. Just cortex is outside, medulla is going to be deeper. So here's the cortex all around here. It's lighter. And then the medulla has the pyramids in it. Yeah? And the pyramids vary in number from person to person. Usually either you have like 8 kind of big ones or up to 15 kind of small ones. Doesn't seem to matter. Like ends up being the same amount of area. This one just has 6. I don't know why. Or 6 at least on this cut. So these are called the renal pyramids, and those are actually in the medulla. There are also these regions between the pyramids. They're called columns. Okay. Those aren't super functionally important in the story that we're talking about, but these guys are. Okay, so just to go ahead and give you a preview of things to come, I'm going to tell you that these 3 processes right here, they start right here at the border between the cortex and the medulla, and they are going to continue all the way until you get down to the tip, and then you're going to be done, and you're going to dump the pee in here, yeah? Gather it up, and then eventually stick it in your urinary bladder. Does that make sense? Okay, so we'll get some more detail about that as we go through today. Okay, so your kidneys are, again, retroperitoneal. They're in the lumbar region. They sit kind of just below your first 2 ribs, right? Okay, and then lower than that as well. They're about the size of a bar of soap. Not dramatically different in size based on the size of the person either. The adrenal or suprarenal glands sit on the surface of them, these guys right here. They're not directly related to kidney function with the exception of like they produce one hormone that regulates kidney function, aldosterone. But other than that, they just kind of do their own thing. They're just like neighbors, not necessarily intimately related to one another. So those are the adrenal or suprarenal glands. The medial surface of the kidney right here, where all the tubes come and go, is called the hilum, that little indentation. Some textbooks call it hilus. That's fine too. This textbook calls it a hilum. Either one is good. Okay, and you're going to see hilus or hilum over and over again too. Anytime you have a whole bunch of tubes coming and going from one place, it's generally called the hilus or hilum. All right, so we already did these layers, and now we are going to do just sort of the internal gross anatomy of the kidney. Okay, so like I said, this is a renal pyramid. This is where really all of the physiology happens. It happens from the border of the renal pyramid down to the tip of the renal pyramid. The tip of the renal pyramid is actually kind of rounded in shape, and it dips into a cup. And anytime anything is vaguely rounded in shape, somebody names it after a boob or a nipple, right? So now you have your kidney nipple, right? Which is your renal papilla, this little guy right here, the renal papilla. So here's the base of the pyramid, and here's the renal papilla, right? And so urine production goes from right out here at the big, like lower portion of the cortex all the way through the base to the papilla. Good? Okay, so that's how urine production is going to go, and we're going to talk about urine production today. And then notice that the papilla is surrounded by a little cup. Yeah? That little cup is called the minor calyx. Calyx means cup. So you got a little cup, minor calyx. And then when you gather more than one little cup together, you've got a major calyx, which is a big cup. Good? Okay, when you gather all of the big cups together, you gather into the renal pelvis, right? And then everything goes from the renal pelvis to where? Make it to the toilet. Ureter, urinary bladder, urethra, toilet. Yeah? Okay, so that's all the basic gross anatomy on your way out. Everybody good? It's not very hard at all. You can actually get all of that pretty easily. Okay, so let's start talking about blood flow through the kidney, and we're going to come back and reinforce urine flow multiple times today. So let's start talking about the blood flow through the kidney. All right, you ready? So with this figure right here, it goes all the way through the blood flow into and back out of the kidney. You guys already told me that you know, of course, that the blood flow coming to the kidney comes from the renal artery. So my question to you is, what vessel was it in right before it got to the renal artery?
So it's
Descending abdominal aorta. Because it's coming right off the aorta, it's going to be pretty high pressure. Does that make sense? Yeah? Still going to be relatively high pressure. And that's going to be important because that's for filtration. Yes? Relatively high pressure compared to a lot of capillary beds. And there'll be other ways that we tweak the pressure as well. Everybody okay with that? Okay. So we are going to go descending abdominal aorta and then renal artery. And then we're going to learn all of these vessels. They sound hard at first, but they're not that bad. What I would do if I were you is, with this trace, which you have to do in lab anyway, right? You have to do this trace. What I want you to imagine is getting a red blood cell into the kidney and then back out of the kidney. Because in a good day, should your red blood cells end up in your pee? No. They should go right in and right back out. But if it was a molecule of urea, yeah, it should go into the kidney at the renal artery and then should end up in the toilet. So we'll do that a little bit later. Right now we're just doing blood flow, right? Okay, so let's do the gross blood flow first and then we'll add in the microscopic stuff. You ready? So renal artery comes in and then it branches. When it branches right here, it becomes what? Those are called the segmental arteries. Yes, segmental arteries. All of the arterial blood flow into the kidney is parallel to the venous blood flow out of the kidney with the exception of the freaking segmental arteries, which somebody decided there wasn't a segmental vein, and I beg to differ, but they didn't let me name things. So watch this, right? Renal segmental, okay? And then I am going to go up the sides of the pyramid. You ready? Up the sides of the pyramid are going to be called the interlobar arteries because they're going between those lobes. Does that make sense? Interlobar arteries up the sides of the pyramid, right? Then it's going to arch around the base of the pyramid, and we're going to call it the arcuate artery. Does that make sense? As in arch? Yes, arcuate artery. Then I am going to give rise to those little bicycle spokes right here. You see them? Yeah? Those little bicycle spokes are not going to be interlobar, but they're going to be interlobular. Does a lobule sound bigger or smaller than a lobe? Smaller, yes. So interlobar up the side, arcuate, right? Then are interlobular arteries. Does that make sense? Okay. Now, from the interlobular arteries, you're going to give rise to what we call the microcirculation of the kidney, but I'm going to come back and pick that up in just a second. Let's just do the gross blood flow out first. So you're going to go up to the interlobular arteries. Then ultimately, you're going to lead to the capillaries, which is where you do all that business, right? We'll come back to that in just a second. After you finish all that business, you're going to go back out of the kidney, yeah? Okay, so back out of the kidney is pretty much parallel with one little caveat. Ready? So we're going to go back out. What do you think we're going to call these veins right here? Interlobular veins. How about this guy, this vein? Arcuate vein. And then what are these guys called? Interlobar veins. And then some yahoo decided that those were not segmental veins. I don't know why, because they should be, right? They just consider them a continuation of the interlobar veins. So you don't have anything called segmental veins, although it would be more logical if you did. They are not called segmental veins. They are just continuations of the interlobar vein. And then it goes into the renal vein, and then into what vessel? Inferior vena cava, yeah? Inferior vena cava. Got it? Okay. Everybody okay with that blood flow? Now we need to do the more complicated part. And the more complicated part is the microscopic blood flow to the kidney. Because of course you can't do any of this until you get to a capillary bed, right? And we were still in arteries during that whole story, right? Arteries and then veins. So obviously there's a capillary bed somewhere. So let's get into it. You ready? Okay, so the microcirculation through the kidney, let's do it. Okay, so what we're going to do is we're going to take this little chunk right here. You see it? Blow it up over here. Okay, and look where we are. This was the arcuate artery, yeah? This is the which one? Interlobular, right? Because lobar is way back there somewhere, right? Interlobular, but the interlobular is going to give rise to a weird looking capillary bed, and we'll figure out why it's weird looking in just a second, okay? So the interlobular artery is going to give rise to this region right here, the important thing for you to know about that region right there is the blood tubes are next to the P tubes. Everything yellow over there is ultimately going to contain urine or something that's pre-urine. Does that make sense? And it's snuggled up to the capillaries, right? So wherever you've got two sets of tubes that are super close to one another, highly branched, pretty porous, right, you can have exchange. Good? Okay, so we'll get to the exchange in just a minute. Let's just do the blood flow first. So my objective here is I took a red blood cell into the kidney and I'm trying to get it all the way back out of the kidney. I'm not trying to push it into the urine. That can happen and you can get blood in your urine. It's not a good day when that occurs. Somebody punches you in the kidney, you can get blood in your urine. You have a massive kidney infection, you get blood in your urine, right? You have massive hypertension, super high blood pressure that makes these holes not this big but that big. Yeah? You can get blood into your urine. Yes? But on a good day, that shouldn't happen. You shouldn't have blood in your urine. So it's a good day today. So let's get that red blood cell all the way in, and we got it all the way to the interlobular artery, right? And now we are going to get it into the capillaries via this vessel right here. This vessel is called the Afferent Arteriole, right? Because it's going toward, yes? It's going away from the cardiovascular system, away from the heart to the capillary bed, Afferent Arteriole. You with me? It's going to go into this weird little ball of capillaries. That ball of capillaries is called the glomerulus. It is going to be super duper high pressure for reasons we will discuss in just a second. Because it's super high pressure, which of these is it going to be really good at? Filtration. That's going to be the primary site of filtration, okay? But even if it's high pressure on a good day, I still get to keep my red blood cell. So it's going to go into the glomerulus, right? And then back out of the glomerulus. Ready? I'm going to go in via the afferent arteriole, then into the glomerulus, and then back out. This is the efferent arteriole, right? Because it's exiting the glomerulus. Afferent in, glomerulus, efferent out, right? Okay. Then when I leave the efferent arteriole, I have two possibilities for parts that I will go through. This picture is kind of confusing because it actually drew both of them. So I'll show you two different pictures about it. When I leave the efferent arteriole, it depends on where this structure actually is. If the structure stays in the cortex, I'm going to go through these vessels right here that wrap around this tube. Those are called peritubular capillaries. And that name should make sense, right? Peritubular, around the tube. Okay, so they're wrapping around the tube, right? But if the structure of this thing actually goes down into the cortex, instead of running into the peritubular capillaries, I'm going to run into these vessels. Those are called the vasa recta, which means straight vessels, right? Okay, so let me show you what that actually looks like with the two different types. Okay, peritubular capillaries, do you see? Yes? Or primarily this is the vasa recta. Why the hell do you care? Well, physiologically, I'll just go ahead and tell you that the whole vasa recta, when it dips down into the medulla, is primarily about water reabsorption. So it's primarily just doing water. This is doing other stuff like ions and glucose and that kind of stuff. So not surprisingly, you have kind of both possibilities in your kidney, right? But this one just has peritubular capillaries and this one has a vasa recta. Okay, they don't actually call those peritubular. Okay, so the thing right here is that those two are at the same point in circulation, right? They're just two different possibilities at the same point in circulation. FYI, if you were a desert mammal, and I guess technically we are, but if you were a desert-dwelling mammal like a kangaroo rat or something like that, you have a really, really long one of these because you do a whole bunch of water reabsorption. Kangaroo rats, their urine is paste, not fluid. Why? Because they reabsorbed almost all of the water possible, right? Does that make sense? Okay. So if you are having pasty urine, you need to have some water. No more coffee. No more cocktails. Water. Water. Okay. I'm not talking to you. I'm talking to me. Okay. Okay. All right. So now let's get the blood back from here, right? So we went into afferent arteriole, glomerulus, efferent arteriole, peritubular capillaries, or vasa recta, and then I'm kind of done with it, right? I've done everything that I could do with the with the blood, and now I'm ready to dump it back into circulation. And the way that I do it is that I dump it back into, what's this guy right here? Interlobular vein, and on my way out. So now let's do the whole story. Ready? From the beginning all the way back. Okay? All right, so I'm a red blood cell. I do not want to end up in the toilet. That's not where they go, right? I'm going to come from the descending abdominal aorta, then into what vessel? Renal artery, and then into what vessel? Segmental. And then go up the sides of the pyramid. What are those called? Interlobar arteries. Arch around the base of the pyramid. Arcuate arteries. Go bicycle spoke from the base. Interlobular arteries. And then from the interlobular, you go into the microcirculation. From the interlobular, you're leading to the glomerulus. What's it called? Afferent Arteriole. And then the little ball of capillaries? Glomerulus. And then away from the ball of capillaries? Efferent Arteriole. And then one of two possibilities. If I stay up in the cortex, what do I call those capillaries? Peritubular capillaries. If I dip down into the medulla, I call them what? Vasa recta. From one of those two, I'm going to go back into circulation, into what's the bicycle spoke vein? Interlobular vein, and then back out, arch around the base, arcuate, down the sides, interlobar, pretend that it's not supposed to be called segmental on your way out, even though it is, right? And then what? Renal vein, right? Okay, so that's your trace through the kidney. It's not actually as bad. So here's how I would practice that if I were you. I would list it all the way out and figure out how many pieces it has, number them, and then you'll know if you're missing one, right? That makes sense? Okay, you'll know if you missed something. Good? Okay, so that is blood flow in and out of the kidney, and we will pick up the actual P tubes in just a second. Let's make sure that we've got all the other stuff too. Okay, do the kidneys have any innervation? Yes. Can your kidneys hurt? Yeah. That means that they have sensory innervation. It is primarily visceral sensory innervation, not somatic sensory innervation. Your kidneys can hurt if you've got kidney stones, get kicked in the kidney, have a kidney infection. They definitely have sensory innervation, right? But then they also have motor innervation. They're hooked up to the autonomic nervous system, but primarily what happens is the sympathetic nervous system just controls the diameter of the blood vessels leading to the kidney, primarily because that will actually predict filtration, right? How much blood you're sending to the kidney is going to predict pressure and therefore filtration. So that's what the sympathetic nervous system does. Don't get confused because it's sympathetic. You just don't have any parasympathetic innervation of vessels. It's all sympathetic with vessels, okay? So, but it's also hooked up to the parasympathetic nervous system, but literally nobody knows what the hell that does, right? You can clip the parasympathetic innervation of the kidney, doesn't seem to make a damn bit of difference. You can stimulate the parasympathetic innervation of the kidney, doesn't seem to make a damn bit of difference. It is hooked up, don't know what it does, right? Neither does anybody else, right? And maybe what happened is it did something in our ancestors and it doesn't do it in us anymore. I don't know. Who knows? Okay. All right, so now we are going to get to the microscopic anatomy of the kidney. We have to do the P part, right? We already did the blood part. So this structure that we've been talking about right here, where the blood tubes are next to the P tubes and you have the opportunity to do that, is called the nephron, or Zach nephron. You'll never forget, right? Zach nephron. So the nephron is the site at which you do filtration, reabsorption, and secretion. You have about 1 million nephrons per kidney. Your kidneys are kind of redundant. You can live with one of them as long as you're nice to it. You can donate a kidney if you want to, but then you can't have cocktails very often anymore. So you got to consider that very carefully, right? Because sometimes it's literally the only thing you're looking forward to in the whole damn day. Okay, so you can live with one kidney. They're redundant. They do more than their job every single day. They're super duper good at their job, and we'll talk about sort of what they do in just a second. So they do this 3-step process, which is filtration, reabsorption, and secretion. Okay. There are 2 major components of the nephron, and we already did the first one. The first component of the nephron is the vascular component, which is the blood tubes, right? Okay, that's where you get everything from, right? And then the other component of the nephron is called, well, it's the P tubes, but that's not what it's called. The vascular component and then the tubular component. So the tubular component, or the renal tubule, I've color coded this for you and drawn it this kind of unpleasant pea yellow color because the common student problem is that they get lost. They start in one tube and then all of a sudden I was a red blood cell and all of a sudden I'm in the collecting duct. So I tried to color code it for you, and then we're also going to do a couple of traces verbally, so you need to figure out which tube should contain what on a good day. So the tubular component, or the renal tubule, is all of the tubes that are going to end up receiving both pre-urine. Pre-urine is called filtrate. I don't care, you can call it urine. And then when it hits the collecting duct, it's actually considered urine, but we'll just call it urine the whole time. Okay, so what is going to happen is I have all of these tubes, right? And ultimately what's going to happen is I'm going to receive stuff from the bloodstream, right, into the tube. I'm going to allow time for reabsorption if necessary and secretion if necessary. But then anything that's still in the tubes by the time I get finished with those three is going to end up in the toilet, right? Okay, so let's talk about the tubular component. And the tubular component is all of those yellow tubes that you are seeing. But the first place that the blood tube comes right next to the P tube is right around the glomerulus. Can you see that? That's the first place that the blood tube comes right next to the P tube. Do you see that the glomerulus is actually sort of snuggled by this capsule around it? Do you see that? Snuggled by the capsule around it, or here? Okay, good, right? So it's snuggled by this capsule around it, okay? That capsule surrounding the glomerulus is called the glomerular capsule, okay? The glomerular capsule is really low pressure. The glomerulus is really high pressure. Filtration pushes stuff from the blood into the pee at that site, okay? Good, if it's small enough to fit through the hole, okay? So that's called the glomerular capsule. They also quite often call it Bowman's capsule. Bowman is the guy who found it. In anatomy, we are moving away from using eponyms, which are proper names for naming. In physiology, all the physiology books still use eponyms. So I usually try to give you like both, right? So you either call it the glomerular capsule or Bowman's capsule. Can you use the terms glomerulus and glomerular capsule interchangeably? No, because one of them in your head should be red and the other one in your head should be yellow, yes? One of them contains blood and the other one contains urine or pre-urine. Yeah? Does that make sense? So you can't leave off capsule and get it correct. Okay? Glomerular capsule. Okay, so what's going to happen is anything that is pushed out of the glomerulus is going to get caught in the capsule, and then we're going to make our way out. You ready? Okay. Make our way out via this curvy tube right next to the glomerulus is called the proximal, because it's right next to it. Convoluted, which means what? Twisty, yeah. Convoluted tubule, which is a small tube. Yeah? Proximal convoluted tubule. Everybody okay with that? Okay. Now, it's hard to see in this picture, but trust me, okay? The proximal convoluted tubule is going to curve, 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 and lead to the descending loop, right? It's the descending limb of the loop. This loop right here collectively is called the loop of Henle or the Nephron Loop. Again, anatomy books use Nephron Loop. The physiology books use loop of Henle. It's the same damn thing, okay? And it goes down and up. This down and up is going to give you a lot of time for water reabsorption, okay? So descending portion of the loop, ascending portion of the loop, yes? Back up to this, which is the distal convoluted tubule, yeah? It's distal because it's further away from the glomerulus, yeah? Convoluted for the same reason. Tubule because it's a little tube, right? So distal convoluted tubule, right? When you get all done with it, you are ready to collect it and dump it into the collecting duct. The distal convoluted tubule leads into the collecting duct. Does that make sense? And it's called a collecting duct because look, it collects from more than one nephron. Yes? Let's go back to the gross anatomy real quick and then we'll do a urine trace. The gross anatomy, notice that these pyramids are kind of striped looking, and the stripes are not exactly parallel. They're kind of converging at the renal papilla. And why are they striped? Because the collecting ducts are all running toward the renal papilla, right? That's why it looks kind of like that, right? Okay, so what I just did is I started urine production up here and I ended it here. And then I'm done, right? Collected it and then I'm gonna dump it out, right? Okay, so here's what you guys, all right? Okay, so we are going to now pretend that I actually delivered a molecule of urea. Urea is a nitrogenous waste product. It's small enough to fit through the holes. You make it when you eat proteins, which you of course need to eat, okay? And it's a waste product that if it increases in concentration in your bloodstream, it can screw up your pH. So you have to get rid of the vast majority of the urea that you produce, right? Okay, and you can't live without eating proteins, right? You actually need amino acids, right? So all of us are going to make urea, and all of us are going to need to get rid of the vast majority of it into our urinary system. Everybody okay? So here's what I want you to imagine, and I want you to do this trace with me. I just ate eggs for breakfast, yeah? Protein, yeah? I broke them down. I had enough time to do protein, the amino acid absorption, and now I've got this urea in my bloodstream from the metabolism of that protein. Good? And I am going to deliver that urea via what vessel to the kidneys? Renal artery, okay? But here's what I want you to think about. When does the urea go from blood tubes into P tubes? So I want you to think about that as we go through. So we're going to pretend that we're a molecule of urea, and we're going to play magic school bus, right? As we go through the kidney. Ready? So I'm in a kidney urea school bus. So I'm going in at the renal artery, and I'm going to do the same damn thing again, only we're going to end up at a different place, right? Renal artery to what? Segmental to interlobar to arcuate, yeah? From arcuate, I go into what? Interlobular artery, so start paying attention. From the interlobular artery, I'm a molecule of urea. I want to end up in the toilet. I am going to go, what's this vessel? Afferent Arteriole to the Glomerulus. And then where am I going to go? Yes, from the Glomerulus to the Glomerular Capsule, right? Okay, I got pushed out because I'm water soluble, relatively small. This thing's high pressure, that thing's low pressure. I got pushed out into the capsule. But I'm a cat turd. Do I want to bring myself back into the house? I do not. So get me out all the way to the toilet. We're going to do the whole damn thing. You ready? Okay. From the glomerular capsule, yes, where do I go next? Stay in the P tubes. Proximal convoluted tubule, and then what? Nephron loop. Which one first? Descending, ascending, and then what? Distal convoluted tubule, and then what? Collecting duct. And now let's go back to the gross anatomy. From the collecting duct, where do I dump? Give me the name of the tip. What's the tip called? Renal papilla, kidney nipple, into your kidney nipple. Little cup, minor calyx, big cup, major calyx, biggest cup, renal pelvis, tube on the way out, ureter, tank, urinary bladder, urethra, toilet. That make sense? Okay, so how do you keep track of what is what? I don't know what will work for you, but you need to find some way that you are really confident about knowing which tube is a blood tube and which tube is a P tube, right? Okay, and conceptually, you kind of have to understand the logic of the system to figure out when you're going to go from one to the other. Does that make sense? Okay, so if I were doing a blood trace again, I would list it out, figure out how many members there were, so I didn't actually miss one. If I were doing a urine trace, you always start a urine trace in what structure? Like which structure is always going to be the beginning of when you produce urine? No, the capsule, right? That's when you first have P, right? So you ended up in the glomerular capsule, and then everything on the way out there is urine. If you're going to stay in the urine, you go capsule all P structures to the toilet, right? You don't go back into the blood, okay? So you have to practice this, right? You just have to list it out. But after you've listed it out like 2 or 3 times, you've got it. It's not as bad as it seems, right? Okay, especially if you color code it. I'm not even a color person. I'm not a visual person, but this stuff has colors in my head, like just to sort of keep track of what's what. Everybody okay? Questions? No? No? Okay. All right, so let's talk about the filtration site in a little bit more detail. Okay, so the site of filtration, not surprisingly, is here, right? Because this is high pressure, this is low pressure. So this is the pitcher and this is the catcher, yeah? If you look at the site of filtration, collectively with the pitcher and the catcher, this thing is called the renal corpuscle. What is the renal corpuscle? It's the glomerulus plus the capsule. Is that okay? Right? It is the site at which we do filtration in the kidney. It's really, really important histologically. It looks kind of like this. If you get like a cross-section of the renal cortex, you can see a whole bunch of those. Okay, so afferent arteriole going in, efferent arteriole going out. Everybody okay so far? Okay, so why does this capillary look so damn weird? Not just the shape, but it doesn't look like the right kind of epithelial tissue. And that is because you're actually not seeing the outside of the capillary. What you're seeing is the inner layer of the capsule. So I want you to imagine that this is the glomerulus, okay? It's of course more branched than this, right? But it's got branches going in and out, right? Okay. The glomerulus, what I'm going to do to put the capsule around it is imagine the capsule is like a softly blown up balloon, kind of like the same idea of the pericardial cavity. I'm going to take that, I'm going to push it around. So what's going to happen is the glomerulus is going to end up with a glove on, yes, right? And then a space, and then the outer layer of that glove, right? So I'm going to have two layers of the glomerular capsule. The visceral layer that's covering the capillaries, then a space, then the parietal layer, okay? That's why that looks weird, because you're not actually seeing the capillary, you're seeing the glove. Does that make sense? Okay? So why are they so closely associated? Because if it's got a glove on, right, everything that I push out doesn't go between the actual glomerulus and the capsule. It goes from the glomerulus into the capsular space, right, which is between the visceral layer and the parietal layer. So the capsular space, guys, is right here. Do you see? Okay. What should be in that capsular space? What's in there? Yeah, really. Yeah, it's called filtrate at that point, but it's basically pre-pee, right? Yeah? Does that make sense? Okay. So this is the relationship. And of course, if it stays in the capsule, it's going to end up in what tube next? The PCT, the proximal convoluted tubule, yeah? Right? Does that make sense? Okay, so I want to look at this in just a little bit more detail to show you why the heck these are so good at filtration. Okay, so all of the capillaries in your body, as we talked about before, do filtration, right? Because the capillary is going to be high pressure, if we're talking about systemic circulation, always high pressure at the arterial end and lower pressure at the venous end, right? So at the arterial end of any capillary bed, you're going to push a little water and some small solutes out. And I told you that's where you get your interstitial fluid, right? Does that sound familiar? Okay. But collectively, all of the rest of the capillary beds of the body filter out about a gallon of fluid per day, collectively. And it's still a lot. But the kidneys, these two, filter out 47 gallons per day. Why are they so good at filtration? Well, there's three reasons that they're so good at filtration. First off, they are high pressure. We already saw that they were directly attached to the aorta, but there's a lot of things that are directly attached to the aorta, right? They have another reason. Just look at the diameter of these two vessels, right? And tell me what it's going to do to the filtration in this vessel right here. Afferent arteriole is big, but the efferent arteriole is small. What does that do to the pressure and therefore the filtration in the glomerulus? Big tube coming in, small tube coming out, right? It's going to bump up the pressure, yes? 1,000,000 times, because you got 1,000,000 nephrons, right, per kidney, or 1,000,000 times per kidney. Does that make sense? Okay, so that's going to bump up the pressure, and that's going to make it really, really good at filtration. And then two other reasons. I have 1,000,000 of them, so there's a lot of surface area, 1,000,000 per kidney. So high pressure, lots of surface area, and then very porous. So if you take a cross section, these are fenestrated capillaries, but they're even more holy than most fenestrated capillaries. In addition to them being fenestrated, the inner layer of Bowman's capsule is made up of these weird looking cells called podocyte, which means foot, like cells with feet, like little octopuses. But between the podocyte cells, there are slits, right? So they're porous as well. This is kind of what it looks like with scanning electron microscopy, and this is an even cooler picture, right? With colorized scanning electron microscopy. So 3 reasons that they're fantastic at filtration. High pressure, lots of surface area, very porous. Yeah? And so they're better at filtration than any other capillary bed in the body by far. Everybody cool with that? Okay. So again, this collectively, both pitcher and catcher for filtration, is called the renal corpuscle. Good? Okay, so we've got filtration slits, okay, proximal convoluted tubule. So the only thing that I want to tell you about the tubules is that this harkens back to something that you learned at the beginning of the semester and probably didn't even know what the hell you were learning. Remember at the beginning of the semester, we said, hey, I'm going to make you memorize all of these tissues, right? And I'm going to tell you that kidney tubules are made of what kind of tissue? Simple cuboidal epithelium. And you literally just bought it. You're like, I don't even know what the hell you're talking about, but I got too much to memorize, so I'm not even going to ask you. Like, what the hell is a kidney tubule? These guys are kidney tubules. And if I do a cross section, they look like this. Yeah? So the proximal and the distal convoluted tubule with a whole bunch of capillaries next to them, snuggled on up with capillaries, right? Those guys right there are the tubules that you learned at the beginning of the semester. And they're special because, of course, you do most of your reabsorption and secretion at the tubules, right? And how are they selective? These cells have a whole bunch of transport proteins on them. They've got glucose carriers on them. They've got ion channels on them. They've got a whole bunch of active transport pumps on them. So they are doing most of those other two, right? So that goes back to the beginning of the semester. That's what we were talking about kidney tubules, right? Good? Okay. All right, so collecting ducts. Okay, so we did the vascular component. We talked about the blood pressure. So now you should be able to label all of these structures, right? All of these structures, and they're all right here. If I were you, I would color code them, but that's all up to you. You can do whatever the heck you need to do. Of course, the test isn't going to be color coded, right? But you have to figure out how to know when you're in the blood versus the pee and which tube you're actually in. Okay, and then one last little thing, you probably won't like it. It's related to physiology. Nobody ever likes it. And every single damn time I teach it in physiology, they pretend that I didn't teach it to him in anatomy. So it's called and the reason partially they don't like it is because it's phys and also because it's got a long ass name. Here we go. This little area right here where the afferent arteriole runs right against the distal convoluted tubule is called the Juxtaglomerular Apparatus. Let's break it down. Juxta meaning next to, glomerular meaning glomerulus, right? Apparatus because somebody liked that word, okay? The juxtaglomerular apparatus is this region right here because in many, not all, nephrons, look where the distal convoluted tubule runs on its way back to the collecting duct. Do you see that? Kind of snuggles up to the afferent arteriole, right? Snuggles up to the afferent arteriole. So the afferent arteriole is going in. It is quite often stuck to the distal convoluted tubule. They're not hooked to each other, but they're stuck to one another. And collectively, those two are going to have an important function in regulating several physiological variables. So let's start talking about them. Okay, so this is a little phys. I know how you guys feel about phys. Okay, phys is way, way more interesting than anatomy, but I'm going to go ahead and give you a heads up that it's harder too, isn't it, Lauren? Is it harder? Yeah, it's harder. Is it harder? What do you guys think? You got through it. Yeah, it's way more thinky. It's way more thinky and way less memorizing. Okay, so afferent arteriole right up against, collectively, these are called the juxtaglomerular apparatus. So I want to do what I do in phys, which is give you the big picture and have you predict what the hell's going to happen. I'm going to tell you that in these two tubes, the afferent arteriole and the distal convoluted tubule, you are going to have the capacity to regulate two different variables, right? You are going to regulate blood pressure in one and the chemical content of the urine in the other. I want you to tell me which tube you are going to regulate blood pressure in. Afferent arteriole or distal convoluted tubule? Yeah, afferent arteriole. There are cells in the wall of the afferent arteriole that are pressure sensitive. So this is important for phys, and for those of you who are about to have a phys test over this stuff, we'll talk about it, and it comes right back around to stuff you just learned. Okay, if those cells are not high pressure enough, they are not stretched enough, they are going to release something which will ultimately do what to your blood pressure? What do you predict? If they are low stretch, they're going to release something that should do what to your blood pressure? Increase your blood pressure, right? Because they're trying to regulate filtration, yes? Somebody tell me why that's a much better place to regulate blood pressure than the efferent arteriole. Why afferent?
Because you want to know before it goes in filter.
Yes, it's pre-filtration, yes? It's pre-filtration. This is the place that you want to regulate blood pressure, okay? So here's what happens with those, okay? I don't ask you a whole hell of a lot of detail about it in anatomy. In anatomy, you need to know that it's actually going to regulate blood pressure. In phys, I'll go ahead and tell you a little bit more detail. What happens is if those are not stretched enough, the baroreceptors or mechanical receptors in the wall of them will release a chemical called renin. And renin will activate the renin-angiotensin-aldosterone system, which will bump up your blood pressure by two different mechanisms. Lauren, will you tell us what those two mechanisms are?
Whoa.
You can do it.
I only remembered it was raining.
Renin-angiotensin-aldosterone system.
Yeah.
What does angiotensin 2 do? I'm going to help you. I'm not going to leave you flailing out here. She's got a test over this soon. She might as
Well,
well.
I just remembered it was low pressure in the kidneys is what signals.
Low pressure in the kidney signals it, and angiotensin 2 causes vasoconstriction systemic, which bumps up your blood pressure, and it causes the release of which hormone, which will cause sodium retention. It's in the name, renin-angiotensin-aldosterone system. Aldosterone. Yeah, aldosterone. Yeah. So it's a complicated physiological mechanism that responds to low pressure by raising pressure two different ways. Okay. So, you're fine. I was just like, I'm trying to make her do it. She won't forget it next time, right? She's going to do amazing on the test because I'm like torturing her.
Wait, what was ADN then?
ADH comes from the pituitary.
No, there's ADN.
Oh,
It was part of that whole thing though.
ANP or ANF, atrial natriuretic peptide factor. Okay, all right, so that's what's going on in the wall of the afferent arteriole. But in the wall of the distal convoluted tubule, that's not a good place to regulate blood pressure. How the hell would that work? What it does regulate is the chemical content of the urine, okay? It's assessing what's going by it, and it says, oh, there's too much sodium here, do reabsorption. There's too much X here, do this. There's too little this here, do secretion, right? That makes sense? So it's doing one last look at the urine before you are done with it, because it's in the distal convoluted tubule. Yes? And if you needed to do more secretion or reabsorption, it's actually more specific than this, but I'll just give you the general overview. If you needed to do more secretion or reabsorption, it will actually cause the release of a hormone, which will impact what's in the urine. It might not impact it at that particular nephron, but it will impact kidney function. So let me just say, if like there's too much sodium going by, cause the release of a hormone, which will cause sodium reabsorption collectively. Maybe not at that one, because that sodium may be already gone into the collecting duct, right? But sodium reabsorption, right, collectively for the body. Okay? So that's the physiological mechanism that occurs with the juxtaglomerular apparatus. And every damn time I ask that in phys, I'm like, do you guys remember the juxtaglomerular apparatus? Crickets. Every time. Just they all pretend they haven't heard. When I know they were in my class. So I'm like, I literally told you that. I know I did. So that's the juxtaglomerular apparatus. Okay. And you should be able to, in this figure, even though it's not drawn perfectly, it's the best one that I could find. In this figure, you should be able to figure out which one's the afferent and efferent arteriole, right? They're not drawn different diameters, but one of them snuggled up to the DCT, right? Okay, and the other one's not. Good. So this is the afferent arteriole, glomerulus, efferent arteriole, distal convoluted tubule. Collectively, what's this thing called right here? Juxtaglomerular apparatus, not crickets. Juxtaglomerular apparatus is what it's called. Okay, it's an interesting regulatory mechanism. But by the time we get to urinary system in phys, my students are like, oh my God, just shut the hell up about the hormones already. I'm like, I'm literally not going to. We've got 5 more weeks. You got to hang in there, man. They're like, no, I'm done. Okay. Everybody okay? Okay. So is there anything that you would like to go through again?
So,
because we did all the hard parts. Go ahead.
can you go back to the picture of the juxta?
You mean Zach? You want to see him? Okay. All right. No. Okay. All right. Where are you? Juxtaglomerular apparatus. Yes. Oh, my picture in the. Yes.
Okay.
Yes, I can.
So can you repeat again? Where's the efferent? Where's the
Here's the afferent, not because they drew it so that it was wider, right? But because it's snuggled up to the distal convoluted tubule. That's how you can tell in that one, right? Because the efferent isn't snuggled up to the distal convoluted tubule.
And those two, the afferent and the
Distal convoluted tubule.
tubule
This region right here.
is the Juxtaglomerular Apparatus.
Perfect. That just sounded exactly right. Go ahead.
So I'm just wondering about oxygen exchange.
Oh, okay. That's a good question. So I mentioned that in the last class, but I didn't mention it in this one. So yes, conceptually, you're doing all of this stuff at the kidneys, but of course the kidneys are a systemic organ and they also need oxygen and nutrients, yes? So as you go through these kidney capillaries, you are also delivering oxygen to the kidneys, yes? And nutrients to the kidneys, and generating carbon dioxide and also waste products for the kidneys. Luckily, the waste products are not problematic because you know exactly where to get rid of them. You're like, it's fine. I'm like right next to the garbage can, right? So you can get rid of them. But do you see that the color actually changes as you go through these capillaries too?
Yeah, but would oxygen be lost during filtration?
It is, but oxygen is lipid soluble, so it doesn't really move much faster at filtration. And then it just moves back down across its concentration gradient. So even if you pushed it out, oxygen is lipid soluble. It just does whatever the hell it wants, primarily concentration driven. Okay? Does that make sense? Okay, and you push carbon dioxide out too, but it comes back in down concentration gradients. So they don't really behave much differently with gases than other things. So you're doing gas exchange and nutrient and waste exchange and all that simultaneously, right? So thanks, kidneys. I'm going to give you some water today. Sure. Oh, look, I did bring water. It always just sits there and tortures me and says, you failed. Okay. Everybody okay? Any other questions? Anything you need to go over again? No? Okay, I'm not going to let you go early, so if just asking questions is fine. You're like, oh, maybe, shh, everybody, go ahead. Did you? No, Jemima? No? Okay, everybody all right? Well, that's the hard part of the kidney, is this stuff, right? Filtration, reabsorption, and secretion is the hard part of the kidney. I'll go ahead and give you a preview of things to come. Physiologically, the primary way that you regulate filtration and change the amounts is just by regulating your blood pressure, right? It's just a pressure differential. So anything you do that really bumps up your blood pressure will bump up filtration. But these are selective, yeah? So, just give you an example of something that you've actually already learned, and you can apply it here. If I, for instance, didn't have enough calcium in my bloodstream, I might need to do more calcium reabsorption, right? Because I know I'd push it out, right? You with me so far? Because it's little, it's an ion, go through the holes. Might need to do more calcium reabsorption. But you already learned the hormone that is related to increasing your calcium concentration in your bloodstream. Do you remember what it is? Calcitonin decreases, PTH increases, right? And so one of the things that PTH does is it targets the kidney to increase calcium reabsorption. There are other hormones that work on sodium. There are other hormones that work on potassium, right? And so what is happening in your body is you're assessing the levels of ions and then releasing whichever hormone can target the kidney to either increase or decrease reabsorption or increase or decrease secretion, right? So basically those last two are mostly hormonally controlled. First one's mostly blood pressure controlled, okay? Now you don't need to know that stuff for now. I'll just give you like a preview for this. Okay, so just with our last couple minutes, so we're almost done, basically. We don't have very much of this. We already did the hard part. So let's just talk about the ureters, and then I'll let you guys go. Okay, so our friends, the ureters, where are you? Okay, so if you look at the ureters are the first place that you are really going to see a preponderance of this tissue that you learned that lines the urinary tract. What was that tissue? Transitional epithelium, yeah? Transitional epithelium starts in the ureters, goes all the way through the urinary bladder and exists in at least the proximal portion of the urethra. In males, it changes type because the male urethra is on average 8 inches long. No, not the penis. I'm not going to try to lie to you people, right? But all the way from the internal structure to the external urethral orifice is all the way about an average about 8 inches long. In females, it's only about 1.5 inches or 4 centimeters long, right? And it stays transitional the whole time. In males, it's transitional, proximal, and then it changes at the distal end. Do you remember what kind of tissue? You have columnar, right? The weird that you only have at the distal end of the urethra, right? You're like, oh, she's talking about histology. I know you guys love histology. That's why I brought it back, right? I heard all of your requests for histology. So anyway, in the wall of the ureters, of course, transitional epithelium. And what kind of muscle do you think you have in here? Yeah, you got smooth muscle, and you actually have 2 different ways because you can move urine by peristalsis. I know it sounds like you shouldn't need to move urine by peristalsis, but you're not always standing up, right? So you've got a circular layer and a longitudinal layer, right? And they move urine by peristalsis from the kidneys to the urinary bladder. And then the outer layer right here is connective tissue because they're actually stuck to stuff as they go by. FYI, last little tidbit about these guys is that because the kidneys, so they average about 10 inches long, but they get longer, right? If your kidneys, like if he's tall, right? His are going to be longer than mine, right? Because it takes a little more room to get from his kidneys to his urinary bladder. So they average about 10 inches long, not dramatically different in length between men and women, just except for body size, right? But they start out retroperitoneal because that's where the kidneys are, right? Right? And they run retroperitoneal until the sacroiliac joint. And even if you don't know what that is before, you know what it is now, right? Sacroiliac joint. And then they pop in, right? And then they go into the peritoneal cavity, right? And then they run and they actually join the urinary bladder on the back side. So they run in and join the urinary bladder on the back side. So here, they actually run along the back side of the urinary bladder, and we'll do this one next time. And then they dump in down near the bottom of the urinary bladder. They run in the back and dump out down near the bottom. So you got your 2 ureter openings and your urethral opening. And then we'll do urinary bladder and all of that kind of stuff next time. So we're done. I will post the, it's not a quiz, the survey for you guys probably within a few minutes. You can do it whenever you want. Your comments are meaningful, so you can rush through it if you want, but if there's something that is actually I didn't ask about, there's a place for you to do free response comments. And I wouldn't ask if I didn't care, because we're actually trying to figure out how to help you guys be successful. Do you know that you guys do know, but did you know that I didn't know? That most of you guys, 70% of my students work, 70%, and most of them work between 20 and 45 hours a week. And that is meaningful. What? I don't know, right? I understand that, but I don't think that I knew that it was that percentage or that many people that worked that much. Which I don't know what to do about it for you except to make more open lab times, right? Put more stuff online. And I know you're going to complain because everybody does. You should reduce the amount of information in anatomy. If I do, then you all die when you take care of each other. So I'm not gonna, right? I'm just going to empathize with how difficult it is. That's all I can do about that. Did I do the survey for Fizz? I have one that I do, but I'm not lead instructor for Fizz. I'm actually trying to get one going, so if I can get my colleagues to join with me, then I'll send it out to them. What kinds of things should I ask about?
Well, I think that it's part because a lot of it